Good afternoon and welcome back, everybody. I am very sorry for the very rushed lunch, but hopefully we will be on time for the afternoon session. I request all speakers to please stick to their time slots so that it doesn't inconvenience the next speaker and we don't have to cut short any of the talks, and that we have plenty of time for questions. With that, I will start the afternoon session. The chair for this session is going to be Karthik Balasubramanian. He is a researcher at he's a faculty at the Agarkar Research Institute, and he will be chairing this session. And the theme of this session is paleomalacology. Sorry.
Thanks, Chinta. Okay, Arvind always give me tough task, and here we are post-lunch session. So without wasting much of our time, we'll go on to the next speaker, Devapriya. Devapriya is a professor at ISER Pune, Earth and Climate Science Department. So Devapriya works on how mollusks respond to chemical changes over evolutionary scales from millions of years to today. And she looked at both fossil and modern mollusk assemblage to interpret environmental conditions. With this short introduction, I will hand over the session to Devapriya.
Am I audible? Okay. And more tricky question, am I visible if I'm here? Probably not. So I think I'll just stand here. Okay. Okay. So we are going to start a session where we are going to talk about probably everything dead. Okay. So I think for a change, it's going to be interesting. Let's see. So I work on mollusks, and I try to look at a larger picture of how things change, especially molluscan community, how things change over different kinds of time, smaller time versus larger time. Because this is not a simple question to ask, although this question has been very. In fact, one of the primary questions that young Darwin even asked, that what changes the community? And the primary factors which might trigger changes has been well understood even back then, which can be primarily categorized into biological triggers and abiological triggers. But the trick is to understand how these changes actually take place over large span of time versus short span of time, simply because what you can observe very minutely with a large database and with very detailed analysis restricts you into an incredibly short time. But then certain processes do not operate in that short time. And the moment you are trying to look at the larger scale of time, your records become really bad. The records become really incomplete. The task is to understand and to balance between these extremely detailed but snapshot of time record versus really not so clear, sort of incomplete, maybe a bit biased, but a long-term record. So is there any way we can balance these things? So that's an overarching question that generally paleobiologists, paleomalacologists are trying to answer. Now, what is this fascination about mollusks? For us as paleontologists, it is not just another group we can decide to choose. In many of the cases, it is the only group we can study simply because of their fossil record. And with that, I try to look at stuff from different perspectives. So students of my group, they look at real-time observation, both in a controlled system as well as in the real marine system. We also do ecological sampling where it's slightly more time averaged. Some has some live animals, but also a depth assemblage. But finally, with all these snapshot time, ecological time, eventually we also try to look at deeper time perspective. Clearly, we cannot answer all the questions, but the primary objective of these research program is to get some understanding of these larger questions and what are the challenges. So once we get all kinds of data from all kinds of time scale, then the eventual work is to basically go through the data, identify bias, and then once you can remove some of the biases, if you can find out biologically meaningful trends which are valid in different time scales. So I'll start with certain things about India. And the goal of this talk is going to be looking at certain biogeographic questions which are related to India, but in different times. So we know that the evolution of the Indian coastline has a very long history, and even the previous speaker in this session have talked about some of these long history of the Indian subcontinent, how it moved. It's one of those very rare instances where a landform actually moved from its polar region, polar location to all the way to the tropical region. Very unusual natural historical experiment that happened. But we are not really looking at that span of time. We are going to start from a time when the present configuration of the seaways have been achieved, and it was just before and after. So just to give you a brief outline of this, so this is the Tethys Ocean. So now if you sort of place it in modern Indian context, where you see Himalayas, there was an ocean at some point of time. And we know that around a time which we call Miocene, the Himalayas actually reached to its present height. So that means by that time, this frontal ocean, the northern ocean, was already closed. But interestingly, this ocean didn't close at one go. It closed in different steps. The front part closed first. So there you have the margin between, in the Jammu Kashmir area, you have these part of Himalayas. But even when the part of this Himalaya is rising up, you have the western part of India near Gujarat, Kutch, that seaway was still open. And that was the last thing which closed even after Miocene. We get a sequential closure all the way to times, which if I translate in a normal language, will translate to something in the tune of 5 million years. So the seaway has been closing ever since. And now, so this kind of shows this progressional path of Indian movement and eventually settling in the present location. So today's seaway looks like this. And if I just go back, the last point where there was a major change, it would be something around 19 million years ago, where the seaway that you see here was continuous and it was actually connecting to Mediterranean. Just for a reference, this is a paleogeographic map. The white color actually shows the seaway and the black color shows the land. And what you can see is this present Arabian Sea was connecting to Mediterranean even 19 million years ago. But around 19 million years ago, a change happened. This is primarily this Arabian Peninsula and Africa, which rotated and closed this sea. And Mediterranean got its own character and getting it separated from the Indian Ocean. So now the question is, this was the last really major seaway configuration change that Indian landmass sort of faced. There has been a lot of work on the biogeographic implication of this closure. There has been work which shows that a lot of mammals migrated from Africa to the Eurasian part, which is very rarely studied, is what is the implication of it on marine fauna. What happens if you completely separate out a continuous chain? If you set out and make two different provinces, do they show differences? are these differences going to be same or different depending on what kind of seaway is surrounding it? Because work in the Mediterranean has shown eventually there has been a lot of turnover in Mediterranean fauna after these separation. But when we look at the Indian part, really anything has been done to put it in that context, that exactly how the tropical fauna responded. There has been ideas that probably the tropical fauna would not show a significant change that you can expect in Mediterranean fauna because Mediterranean is a closed ocean and the ranges are going to be shorter and hence they are more prone to show changes. But probably in tropical fauna, we are not expecting it. Again, all these are ideas, it needs to be tested. And that was the primary reason why we selected a spot here, which has a good fossil record, which spans from this time. And primarily, we have times before and after. And this is the spot where we collected our fossils. A little bit about mollusk shells when you find in the field and how you associate with some boulders, maybe waves, and you get these interesting mollusks, especially for the marine world. And this is how the field looks like for us. It's completely barren. It has some sparse vegetation, and every point that's sticking up, each one of them is a fossil, and it's a marine fossil. So we walk on this seafloor, exploring all these shells and reminding ourselves that the present seashore is miles away. Still, we are actually experiencing the seafloor with its entire fauna present. The second challenge is, how do we figure out what age is it? So there are different techniques that we can employ, but one point to note, that's another fascination for mollusks. Mollusks have these calcium carbonate shells. The calcium carbonate is precipitated from the seawater where they were living. That means they are also extracting the seawater chemistry of that point of time. And using those shells, you can actually backtrack those seawater chemistry. Considering the biology focus of this general group, I'm skipping that part, just leaving with this idea that there are two things that we can do using fossil molluscan shells. One is looking at their specific isotopic ratios, which tells us something about the seawater chemistry. And certain isotopic ratios also tell us something about their age, not how young or old they were in terms of their own age or ontogenetic age, but more about what age did they get fossilized, so the age of that trigger. That helps us in terms of reconstructing which spot we are collecting from and age-wise where do they fall. With that, we were basically collected a lot of fossils, and we found 3 formations. These geologic formations indicate times before, during, and after the seaway closure. And our goal was to understand how groups changed across this boundary. So these are some plots, some photographs of some of these fossil mollusks. Clearly, it's not as pretty as the fresh mollusks you get in the sea beach or in the live shelf. But believe me, we get more excited to see it than a fresh shell on the beach, because each one of them can give you this unique proxy of seawater chemistry, the time, as well as their morphology. And morphology can change as a response to these climatic changes. So we looked at individual valves, and I'm primarily talking about bivalves, representing 15 families, not a very diverse thing. And it's not unusual to see it simply because not everything gets preserved. If you think about bivalve shell compositions, all of them are calcium carbonate, but their minerals are different. Some of them are called aragonite, some of them are calcite, and both of them do not have the same preservation ability. So certain groups are slightly more resilient to dissolution. Those are the groups that we find. The other groups are slightly less, they have a less preservation potential. So considering all of these bias, we are still happy that we get these panels. And eventually we try to look at their body size, the diversity pattern, and also their community composition. And when I say community composition, I primarily am talking about the species composition. If I look at different biogeographic provinces, are they changing? Are they the same? And we are using traditional taxonomy because very few of them still persist today, and therefore limiting the molecular taxonomic approach fairly restrictive. So it's only traditional taxonomy that. So what we did is looked at different size parameters and tried to see if there is a standard trend. And we did not really find it. So next thing that we tried to do was test this idea that especially for body size or even for the diversity, are they showing a trend in terms of the change? So we tried to test certain models in terms of a maximum likelihood analysis, some of the evolutionary model, that is it, can we explain it in terms of a random change, or is it a directed trend, or overall it's basically not changing? And what we found eventually is it's all of these things, be it the richness, be it different kind of size parameters, it overwhelmingly supports this evolutionary model of stasis. It basically says that it's not changing over time. And this is in stark contrast with the Mediterranean side, where almost around the same time, we are seeing turnovers, we are seeing extinction. So this is one of the clues which says that the tropical ecosystem, especially the ones which has large seaway connectivity, are not responding the same way what a relatively small and restricted basin is going to change. And that, I think, eventually we should also keep it in mind when we are predicting how fauna is going to change. It's not the same, even if they are subjected to the same environmental factor, probably the connectivity makes a big difference. But here it's difficult to predict that just from this. Now, from this point, I'm going to take a few steps ahead and going to look at some time which is much closer to the present. The first question that I was trying to understand is, since then, after this changeover, the coastal morphology did not change much. The seaway connectivity did not change much. How much do we see, and what are the patterns, what are the drivers that finally decide on which groups are where, and study to only bivalves, because again, they are preserved, they are found almost everywhere, and considering the expertise in our group, we can identify fairly easily. That always is a good thing. So the next step was to look at the relatively modern day distribution. Now, when I say relatively modern day, it comprises of the shells which are not fossilized, but they are not fresh shells or like live shells yet. So we are looking at a dead shell. Now, again, the question is, why are we interested in dead shells? Because finally, we want to see a stable baseline with which we can then compare the modern living organisms. And this baseline is very difficult to establish, because if you are collecting only the live specimens today, it may have already gone through certain changes in past 10 years, 15 years. It becomes very difficult unless we have an historical ecological collection. And I think in some of the discussions, probably we will bring it up that is the need for that, and the fact that we don't have it. In absence of that, the best possible thing that we can target is the dead shells. And it's not just this part and certain things about India, which makes it really an incredible India in terms of testing certain influence of different biogeography, different morphology, coastal morphology, is that these two coasts of India, which are placed almost in the same latitudinal band, are very different in many of the aspects. Some of the aspects include that in the East Coast, you have these largest river systems, and you have extremely high siliciclastic input, high freshwater input, and seasonal fluctuation in salinity is extremely low. You go to the next West Coast, it's only a few rivers. The shelf width is different from the East Coast. It has almost nothing comparable to the East Coast river freshwater input. So this kind of makes it this interesting region where you can try to understand over a long term what guides the distribution of organisms, especially if I talk about the bivalves, what guides their distribution in these shallow coastal regions. And that's what we try to do. And again, as I'm saying, that sampling is always a big issue. If we are really trying to understand that over a long term, in the sense of a time average sample, what is the pattern that they are giving. And that's why when we decided to collect things from these locations, there are quite a few, we went for, first tried for live assemblages, then we realized that it's very difficult if we are trying to do it throughout the thing. And we tried to look at the literature, which always argues that if you are looking for coastal region, and especially in the shelf region, you can expect to get a good resolution. Sorry. I'm just going. Okay. Yeah. So this is the pointer, right? The top one is the pointer. So we decided to go for a depth collection, which is basically a bulk collection from the beach or nearby areas. We will scoop out sediments and in a grid and take all the shells that we can find in that grid pattern, and we will repeat this exercise again and then eventually identify them and post to our repository, and this process continues. So after a long time, eventually we managed to get a decent collection from all of these places, which gave us for the first time some general understanding of how distribution is guided. So when we tried to look at the distribution of species, and what we were trying to figure out is, if we look at the species similarity, how these different spots are located. Are they completely random, or do they have a coast-wise divide? That if we plot all of these specific species composition in a grid, is all the East Coast ones are located in the same region versus the West Coast? When we try to do it, we basically found that there are slight differences in the diversity between East Coast and West Coast, where you find that the East Coast is relatively species rich. And to the contrary of our expectation that East Coast and West Coast are going to show you a very different signature, it's not. It's actually the northwest part, which appears to be a biogeographically isolated region compared to the rest of the Indian region. And this, at the same time, was very surprising to us because we didn't expect it to. And when this was work in progress and we found this thing, coincidentally, this was also the time when I started finding out some of the discoveries that Bharti mentioned yesterday, that their genetic networks are also kind of showing similar patterns where the northwest are showing some slightly different patterns compared to the southern part of the west coast. And we try to do the same analysis, not just with our own data of these beach collection, but also literature-based data. Now, the problem with this literature-based data is that when somebody reports an occurrence of a particular bivalve shell from a particular region, especially in Indian context, often it's not very clear whether it's a live shell or it's an empty shell. And this is generally has been regarded as an empty shell problem because the empty shell could be 1,000 years old and you have no way of knowing. So the occurrence level data, if we compile, and we did based on the literature, it also shows the same pattern. So there can be two things which can be an interpretation of that. One is that the live organisms are also showing this, or that most of these occurrence data that has been reported are based on empty shells just like us, and therefore there is such a overlap of this. And it's yet to be explored that which one of this is true. But going forward, it kind of created this thing, and then we also tried to understand individual grid size and what are the environmental parameters or oceanographic parameters that are working in each of them, and can we explain the species distribution from that. It was not very clear, but overwhelmingly the salinity mean and the salinity range showed a pattern that tend to control the distribution of the species. Now, some preliminary understanding that we were trying to get our handle on is why is the East Coast more species rich compared to the West Coast? Again, it was not very well documented before. One aspect of it, which we find even support from the literature, that the East Coast of India, if you look at it, it's actually well connected even to the biodiversity hotspot. There has been some exchanges. On the other hand, the West Coast of it's actually there has been a biodiversity hotspot in Mediterranean during the time that we were talking in the fossil record, but none since then, and it is not connected to. But this East Coast connection probably continued for a very long time and contributed to retain this high species diversity. The other very specific reason for West Coast to be species poor is the fact that in West Coast, we do have this oxygen saturation limit. So there are places, especially the north of the West Coast, where the oxygen concentration seasonally fluctuates and it can go to very low levels. So that's one point which may influence some of these diversity factors, and only specialized group can survive there. So this oxygen limiting probably guides overall species. Now, when we try to look at the contribution from the nearest biodiversity hotspot, again, based on literature search, it shows a difference. So it's not completely understood that why East Coast is still high diversity. It's not like all the species are common. They have their own endemic groups. But this is an open question which still needs answering, that why, unless it's just because the West Coast diversity is low, in comparison, East Coast is high, we really do not know at this point why there is such a difference in terms of diversity, irrespective of which latitudinal position you are looking at. The divide where we found a disconnection in terms of species composition matches very well with the salinity divide. So this is a plot of how the salinity in the Indian Ocean changes in the different seasons. So what you see is during summer, there has been a lot of freshwater influx, and this freshwater influx kind of makes it all the way to this point, but not beyond this. In the winter also, there has been changes, but again, the changes stops right around this point, and therefore the northern Arabian Sea is always this place which does not receive enough fresh water, and it's highly evaporated. So when you are not receiving enough fresh water and the evaporation continues, it becomes a high salinity zone. And this high salinity zone only caters to certain type of species. And we think that is the barrier that we are seeing in our species composition map, which makes this part completely different than the rest of the Indian provinces, biogeographic provinces. Moving onwards, we also thought that it is a good idea to look at some of these responses in terms of individual body size. And why not do more complicated shape analysis or any other morphological trait? Very simple, we had 20,000 shells, and easiest thing that we can do is to measure them. That's the easiest measurable criteria. And literature to demonstrate those are not really bad criteria. Body size in a larger scale controls a lot of metabolic things, and therefore you have a large idea about the body size preferences or body size changes. It could at least guide you to certain direction where you can figure out whether there is at all any change or not. And that was the approach that we took. So we measured the shells all along, and we tried to understand that which oceanographic parameter is controlling them, and overall, is there a change? So we again looked at possibly just plotting them in all possible measures. And if you look at it, clearly there is no trend. I'm not even going to claim that there is a trend. It's all the same. But things slightly change when we look at only the pooled one and only the mean differences. Then it somehow shows that the northwest is slightly larger compared to the southwest. Okay? Again, around the same divide. So there has been a body size change. If we do it for all kinds of families, certain families show up to show this difference to be significant. Other families are not showing it. Then the next step was, is there any ecological factor which is controlling which groups are going to be sensitive and which groups are not going to be sensitive? So therefore, we looked at these two different habitats. So certain bivalves are epifaunal, they live above the sediment substrate, and some bivalves live inside the sediments. And these are epifauna and infauna. We simply wanted to see if the body size difference that we observed are the same in epifauna as well as in infauna. So when we tried to look at that, we found that the epifauna, which are living above, they almost show no difference in terms of the body size. And this completely changes if you look at the infauna. The infauna actually do show a size difference when you are looking at the northwest versus southwest. Now, what might be driving this? So there is no specific study that has been done in these regions, which tell us something about their physiology, which might be controlling. But in general, there are global literature which says that the epifaunal groups, they're much better at handling the environmental stress. And often what we find, so these are also related to some of these oceanographic parameters that I already talked about. So what has been claimed that in freshwater, when the freshwater influx is more in the marine system, the epifauna often maintain a steady energy intake by changing their ingestion rate, producing more pseudospecies. But this is not found in infauna. So with high freshwater input, it is expected that the infauna is going to show the change in terms of their body size, whereas the epifauna is constant. Now, the other interest that we have understanding this is if we can really establish it as a proxy, as a physical response, physiological response to fresh water, then we can use it for the fossils. Because in the fossils, you measure the body size. And the timing that we are looking at also coincides with the timing when Miocene monsoons started. So if we can establish this as a potential proxy for a freshwater input, then we can take this and apply it and understand what was the response of this entire fossil community to the change when the first intensification of monsoon happened. Because monsoon was not there throughout. It was only during the Oligocene-Miocene period when it started. So can we actually trace those starting of the monsoon? So I think in summary, what I'm going to try to say that I'm very glad that Arvind invited me. And I'm even more glad that I came here and interacted with all the real malacologists. But I don't feel out of place because probably with the introduction of Godwin Austen, who was a geologist and who was a paleontologist, who was a malacologist. In fact, if we go back 200 years ago, we are those group of people whom people used to refer as naturalists. These subdivisions are made very recently. And often these subdivisions are so pinpointing on the specialization, we often feel or we often lose track of the larger questions, which are inherently multidisciplinary. So please bear with some people who are not very good at maybe taxonomy, maybe they are better at identifying fossils. Maybe there are others who are extremely good with their geochemistry skills and inferring about the environmental relevance, but really bad when it comes to picking out fossils. But if we are really targeting for larger questions, and many of these biogeographic questions are large, they are multifaceted. They cannot be answered just by looking at which groups are present and which groups are absent, even just by looking at molecular phylogeny. This is the best forum to target some of the large questions and to find out expertise which can complement each other to answer those questions. Eventually, we start with things which are very pretty and very clear, but it's of a small temporal scale. And not all the questions can be answered into small temporal scale. Then it gets blurred, but you get a slightly longer time scale. You go to fossils, it's completely blurred, but it gives you this ideal temporal scale where biogeography actually changes things, where groups evolve in really large time scale. And therefore, I think a combined approach is always the necessity, and what best place to do it apart from a malacological conference where even the whole session is full of paleontologists. Okay. Again, thank you for giving me the option. This is an approach which is kind of inverted, that where for a very long time we thought that we have to understand the present day to understand the past. But now we are coming back full circle that unless we understand the dynamics over long term, which is coming from sort of paleobiology or fossils, even the future conservation efforts would be restricted only by our understanding of very short scale processes and might not be complete. This is an example from California where they have resurrected some of the vulture species which were near extinction using the data from the fossils that what was the habitat like for those extinct vultures. So hope for the future is actually a better integrated approach of conservation biology, which is a continuum. And all the evolutionary processes, as we know, they are not discrete, they're continuous. With that, I would really stop and thank you and be happy to take your questions.
Thanks for the wonderful talk, Devapriya, and also special thanks for completing 4 minutes before the time.
Loved your talk. The northwestern coast, you talked about that turnover in the northwestern coast. The species composition is quite different. Now, is the species composition there more similar to what you find in the coastal regions of Middle East, for example?
No, so that's the other thing. So, oh, well, I have to put it. So they are very different from, let's say, Mediterranean. Okay, but when we come to, let's say, Iran and Middle East, one problem is that we don't have, I mean, very good data points from all the regions. So comparison becomes difficult. But if there are very good work from Oman coast, and many of them share similarity with Oman, although they are kind of way across. So the way to disperse probably would be all around the shelf.
Yeah.
So I'm guessing that they will probably have similar composition.
Yeah, because the salinity seems to be exactly
Yes,
the same.
Yes,
Yeah.
yes. And also they also face this oxygen, a lack of oxygen or deoxygenation. So
Thanks. Any other questions? Thought of announcing a special prize for asking question in this session. Since nobody is asking questions, so that prize I will keep it myself.
oh, are you going to ask me a question?
No.
Okay.
So if there are no questions.